Okay, so good evening, everyone. Today this is an extra problem-solving session for this NPTEL course, Molecular Biology. So as I mentioned in the last week that I will cover the topics of CRISPR-Cas and TALEN in this problem-solving session. So today we will be mainly dealing with these two topics, and tomorrow there will be another session, which would be the last session for this course. The CRISPR-Cas9 and TALEN, these are the two genome editing tools which are used in genetic engineering. So, uh, so these topics were covered very elaborately in the lectures. Uh, so, uh, we will solve a few sample questions that will help you further brush up your concepts. And if you have any doubts, we can discuss that as well. Okay, so I will quickly share today's slide. So today's is a 1 hour session. Share today's slide. Okay, good evening, everyone. I'm Akash Jyoti Pathak. I'm one of the PMRF TAs for this NPTEL course, Molecular Biology, and I'm a PhD scholar in the Department of Biosciences at IIT Bombay. This NPTEL course, Molecular Biology, is being run by Professor Vishal Trivedi of IIT Guwahati. So, the topic of today's discussion is mainly these two genome editing techniques. One is the TALEN and another is the CRISPR-Cas9. Uh, so I'll start with CRISPR-Cas9. So this is a very new technique if compared to what we have been discussing, like blotting and other techniques which were discussed. So compare relative to that, this is a relatively new technique, and so let's start what this technique is. This technique discovered by was discovered by Dortna um, and one of his colleagues, one of our colleagues. And for discovering this system, she was also awarded. The group was awarded the Nobel Prize. Uh, so so CRISPR-Cas system is it is basically a defense mechanism of bacteria to it is a defense mechanism by which in bacteria prevents or attacks or protects itself from attack by bacteriophages. So if you see, this is a bacteria. So there is a specific sequence which is present in the genome of bacteria, and this specific sequence, which is characterized by repeats and spacers, or alternately, this is called a CRISPR array. Okay, so what does this CRISPR stand for? It stands for clustered, regularly interspaced, short palindromic repeats. So as you can see in this figure, so this is the particular genomic or the region of genome which is having this CRISPR sequence. So here you can see that it is so as the name says, it's a clustered, regularly interspaced, short palindromic repeats. So these are, you can see here, these are as been labeled. They are the palindromic repeats, and this S stands for spacer sequences. So we will learn one by one what is the meaning of this R repeats, and what do this S sequences are. What are the spacer sequences? So. So this CRISPR consists of a series of regularly spaced short repeats in the bacterial genome. So these are specific sequences that that are repeated multiple times, and they are present, or the spacer sequence is flanked by these repeat sequences. So the CRISPR sequences and the Cas protein are components of the immune system that have allowed bacteria to survive infection by bacteriophages. So there are Cas genes which will code for the Cas protein. Okay. And there is this CRISPR array. This CRISPR array, give me a second. Okay, so basically, I'll just give up. I will discuss these things in detail. So just to give an overview, these spacer sequences are the sequences of the phage genome. So whenever a bacteriophage attacks a bacterium, a part of its genome is 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 incorporated in the bacterial genome at a specific loci or specific location. So these spacer sequences that you see, they are actually part of the viral or the phage genome that have been cut from this phage genome and incorporated. So it basically acts as a memory system. So the memory system to so that if that bacteriophage again attacks the bacterium, it can prevent its attack by this CRISPR-Cas mechanism that I'm going to discuss just now. Okay. So, okay. So, it's based on this concept, answer this question. So, CRISPRs consist of conserved repeat sequences interspersed with dash. So, just now we discussed that that if you see a CRISPR array, so it is a particular genomic sequence which is present in most of the bacteria. So, they have palindromic repeats, and these palindromic repeats are interspersed. This palindromic, they are interspersed with what? They're interspersed with spacer sequences. Yes. So these are the spacer sequences. Spacer sequences are the sequences of the phage genome, okay, which have been incorporated in the bacteria. Okay. So yes, answer is option number C, short spacer sequences. Okay, so this as was also discussed in the lectures also. So this CRISPR features short conserved repeated sequences. Just give me a second. Okay, so this concept was discussed in the lecture as well. So these are short conserved repeated sequences interspersed with similarly sized spacers. And these repeat sequences, the length of these repeat sequences ranges from 23 to 47 base pairs. And the spacer sequences range from 21 to 72 base pair. Where bacteria feature multiple CRISPR loci. So there can be multiple locations, where this CRISPR sequence is present, may be present, and each with diverse and hypervariable spacer sequences, even among closely related strains. So these spacer sequences are hypervariable and these unique. So the spacer sequences are unique and they originate from the viral or plasmid DNA and serve as the recognition elements. So these spacer sequences will be recognized by the Cas9 protein. Okay, and how it recognizes the sequences, we will understand in the subsequent slides. The introduction of new spacers allow the system to recognize and eliminate matching viral or plasmid genome. Okay. So this is the Cas gene cassette, which will code for the Cas9 protein or the Cas protein. There are different types of Cas protein, and this is the CRISPR array. repeat spacer array, which is the spacer sequences, and which are flanked by the repeat sequences. Okay. And you can see over here this figure, it has shown in that this particular sequence of the phage or the phage DNA, this particular sequence, which is called protospacer, it has been incorporated in this CRISPR spacer array. So it is actually keeping a record, or it is keeping a memory of what all phages have attacked the bacteria. So these sequences, the spacer one, spacer two, so it is arranged in a chronological order. Means if the most recent bacteriophage that has attacked the bacteria will be spacer number one, and then followed by spacer number two, spacer number three. So it is a record of all the phages that has attacked the bacteria. Okay. So here also the nucleotide sequence of the spacer must be similar to a region in the phage genome called as protospacer. So when this sequence is present in the phage genome, it is called protospacer, and when it is incorporated in the bacterial genome, it is called the spacer. Okay, so the viral sequences are basically they are what? They are the spacer sequences separating the CRISPR sequences. When the same bacteriophage again attacks a bacterium that is corresponding CRISPR-Cas system, the CRISPR sequence and Cas protein act together to destroy the viral DNA. So first the CRISPR sequences are transcribed. So what is the process? So this CRISPR sequences, they are transcribed to RNA, and individual viral spacer sequences are cleaved to form products called guide RNAs, which include some adjacent repeat RNA. So you saw the CRISPR gene. So in the CRISPR gene, there are alternatively the spacer sequences and the repeat sequences are arranged alternately. So from this gene, what will happen? First, the pre pre pre-CRISPR RNA. This is named as pre-CR RNA is transcribed. When this pre-CR RNA undergoes some modification to give us the modified or processed or mature CRISPR RNA. And so the mature CRISPR RNA will have a spacer sequence, and it will also have a tracer RNA sequence. Okay. So it is transcribed to individual viral spacer sequences. And together, this entire unit, this is called the single guide RNA. Okay, so there will be a spacer sequence, and there will be a this is actually a tracer RNA. But the tracer RNA or transcribed cRNA basically, not synonym is sounding as tracer RNA, but this is transcribed cRNA. So this acts as a scaffold. This acts as a scaffold, and this transcribed cRNA. This is recognized by the Cas9 protein. So together, this Cas9 protein and this RNA form a ribonucleoprotein complex, which goes and binds to the particular or the target site of the DNA, which has to be edited. Okay. The resulting complex binds specifically to invading bacteriophage DNA, cleaving and destroying it through nuclease activities associated with Cas proteins. Okay. So we will understand how this happens. So CRISPR-Cas9 is a type of dash system found in bacteria. So it is an immune system, right? It is an immune system of bacteria. Okay. Yes. So CRISPR-Cas system, they are sophisticated adaptive immune system which are employed by prokaryotes, and CRISPR is a DNA sequence found in prokaryotes like bacteria and archaea, originating from previous bacteriophage infections and serve as an adaptive immune system. So it is an adaptive immune system which is present in the bacterial genome, and it allows the cells to detect and eliminate similar DNA threats. So around 50% of the bacterial genomes have these CRISPR sequences, and nearly 90% archaeal genome have CRISPR sequences. So they are conservatives. Okay. So CRISPR-associated protein, Cas stands for CRISPR-associated protein. It is an enzyme that uses CRISPR sequences as a guide to recognize and open up specific strands of the DNA. So this point we will understand in the next slide. So let's understand first how this mechanism of CRISPR-Cas operates in bacteria, and then we will understand how this has been utilized, or is being used in for genetic engineering applications. Okay, so this figure is taken from this website. So the illustrations and illustrations have been taken from this website. So here what it has been shown that this CRISPR system basically works by remembering the phage infection. So the first step of CRISPR immunity is making a memory. Okay, so this is a phage which has attacked a bacteria, and when the phage infects a bacterium equipped with a CRISPR-Cas system. So this is a bacteria. So this bacteria is having this CRISPR-Cas system. Then a new spacer sequence can be added to the CRISPR array. So this is the bacterial genome having this CRISPR array sequence, and in this CRISPR array, which is present in the bacterial genome, this particular region, a portion of the genomic portion of the fudge is incorporated into this CRISPR array. So you see over here, so as I was saying, that these spacer sequences, they are arranged in chronological order. So starting from the first spacer sequence is the sequence of the phage, which or the sequence of the most recent phage. And subsequently, the other species sequences are the older phages which has attacked the bacteria. Okay, so this system incorporates a new repeat in the process. So all spaces are always found by repeat on each side. So each spacer you will see there. So these repeat sequences are palindromic. So that is why they are called this palindromic term in this CRISPR name. Okay, these are palindromic. So so remember, so look at this figure very carefully. So this is the bacteriophage. So the bacteriophage is injecting its genome into the bacteria, and this Cas1, Cas2. This is a protein complex which is present in the bacteria, and this protein complex helps in recognition of a particular sequence of this phage genome. Okay, so this has a particular sequence known as PAM sequence. So this particular region is recognized by this Cas1, Cas2 protein complex, and then this protein complex integrates this genomic sequence into the CRISPR array, which is present in the bacterial genome. Okay. Okay, so this is called the acquisition phase. So acquisition means this particular genome is acquired in the bacterial genome. Okay. Next step is what? It is expression. So expression, by expression, what will happen? This CRISPR gene sequence that is present, that will be transcribed into a precursor CRISPR RNA. Okay. So these are the, as you can see, these are the spacer RNAs that have been transcribed from this array, and these are the transcribed cRNA. So these are called the tracer RNA. cRNA. Okay. So expression, what will happen? There will be CRISPR RNA biogenesis. So expression means CRISPR RNA biogenesis. It involves transcribing the whole CRISPR array of repeats. So whole CRISPR this array is entirely, this array is transcribed as a single unit and spacers into a long piece of RNA called as precursor CRISPR RNA or pre-CR So this entire CRISPR array is transcribed as a single unit, which is called the precursor CRISPR RNA, uh, and then this Cas proteins, what will happen? These Cas proteins will cut or cleave the long RNA into short individual segments. So the Cas proteins will come and they will cleave it into short RNA sequences. So these RNA sequences, short RNA sequences, the processed RNA sequences are the CRISPR RNA. One is the precursor CRISPR RNA and another is the CRISPR RNA. So the CRISPR RNA is the modified RNA. So here you can see it is having one spacer and parts of the repeat. So this is the part of the repeat. So since these are palindromic repeats, when this will get transcribed, it will form a hairpin-like structure, secondary structure. Okay. So this CRISPR RNA will have two parts. One is this spacer sequence, and another is this repeat. The repeat, when it will get transcribed, it will form hairpin-like structure. So the final full trimmed RNA copies of the phage memories are called the CRISPR RNAs. So these are the trimmed RNA copies of the phage memories are the CRISPR RNAs. Next, the Cas protein. So that these Cas proteins will then come and form a complex with this with this RNA particles and positively identify matching phage DNA. Okay, so the final step in CRISPR immunity, what will be the final step? So once this complex has been formed, this Cas9 or the Cas protein and this RNA has formed a complex which is called the Cas RNA complex, Cas RNA complex. Then the next step will be destruction of the invading fast DNA. So how does it destroy the invading fast DNA? So basically, this spacer sequence that you see, that has a sequence which is complementary to the sequence which is present in the genome of the attacking bacteriophage. Okay, so you see the it forms a it the this sequence, this spacer sequence forms um, it forms a hydrogen bond with this genome of the virus, and then this Cas protein, it is an endonuclease. Okay, it is a nucleus, so it will cut these regions of the or it will cut the genome of the bacteriophage and leave it into very short pieces. Okay, so this process is called interference because it involves the CRISPR system interfering with or stopping the phage's life cycle. Why it is called interference? Because it is stopping the phage's life cycle, or it is interfering with the phage's life cycle. So when a phage injects its DNA into a bacterial cell, the surveillance complex scouts the phage genome for a sequence that matches the spacer in its CRISPR RNA. Okay, so this target sequence, when it will match with the sequence of the spacer sequence which is present in the CRISPR RNA, then these two will form complementary bonds to each other, or it will form hydrogen bond with each other. And what will happen? This Cas9 protein will cleave it and chop it into and degrade this part of the genome, which can further be degraded by other endonucleases and exonucleases present in the bacterial system. Okay, so the Cas1 Cas2 complex plays a vital role in which phages. Okay, the Cas1 Cas2 complex play a pivotal role in which phases of the CRISPR-Cas system. So basically, it is in this question, what is asked is the Cas1 Cas2 complex is playing a role in which step of the bacterial infection. Or which stage of the CRISPR-Cas system, which is taking place in the bacteria. So the answer is what? It is taking place in this. So there were three phases. One is the adaptation or acquisition, not adaptation. First one is the acquisition, second one is expression, and third one is what? It is interference. So the Cas1, Cas2 complex was involved in the acquisition phase of the CRISPR-Cas system, right? So that is the answer is option A. Adaptation is also called acquisition or adaptation. So both are the same thing. It is either adaptation or acquisition. So in the adaptation stage, you can see this Cas1, Cas2 complex acquires a protospacer from the invading. So this protospacer is a sequence which is present in the invading phage genome and integrates it as a new spacer. This is being integrated as a new spacer into the CRISPR RNA. Next question, in the adaptation stage, the dash complex acquires a protospacer from the invading viral DNA and integrates it as a new spacer into the CRISPR array. So in the adaptation stage, which protein complex is involved? It is Cas1, Cas2 protein complex. So Cas1, Cas2 protein complex acquires a protospacer from the invading viral DNA. So answer is option B. Now in the interference stage, interference is the last stage, right? The dash complex identifies the invading target DNA through the complementary base pairing. So the complementary base pairing will take place between what? It will take place between this ribonucleoprotein complex. So it will take place between the CRISPR RNA Cas ribonucleoprotein. Okay. So the CRISPR RNA, which was transcribed from the CRISPR array, which was present in the genome of the bacteria, and after this entire CRISPR array is transcribed as a single unit, and then this is processed. Then this is processed to form the CRISPR RNA. This CRISPR RNA and the Cas protein form a ribonucleoprotein complex. Okay, so the answer is option number B. Okay. So the Cas12a gene codes for an endonuclease that uses the dash to find and cleave viral DNA. So any Cas gene, what will it code for? So the Cas gene will code for what? It will code for a Cas protein, right? Protein, and this Cas protein is inactive. So on its own, the Cas protein is inactive. It will need a RNA molecule. It will need a RNA molecule to so the RNA molecule or the CRISPR RNA, when it forms a complex with the CRISPR RNA, then only it will know where is the target site, where is the target site in the DNA where it has to bind. Okay, so that uses a spacer sequence, right? It uses a spacer sequence to find and cleave viral DNA. Okay, so the spacer sequence is providing the specificity, or it is telling the Cas protein where it has to bind in the viral genome, or or what is the target site of this Cas protein. So that is um, that is defined by the spacer sequence. Okay, the answer is option. Uh, okay, so spacer, not the spacer sequence, this total sequence, spacer sequence together with this tracer RNA that I was saying, transcribe cRNA, this hairpin-like structure together, this is called the guide RNA. Okay, guide RNA, single guide RNA. So answer is option D, not spacer sequence. Now spacer sequence and together this, because this region, this tracer RNA will be recognized by the Cas protein. Okay, so it is acting as a scaffold. Okay, so let's understand this one quickly. So if you see the Cas9 protein, so this there are many there are basically two classes of CRISPR, and in each of these classes there are types and again subtypes. So we will just understand the so these all types and classes have been covered in your lectures, but we will understand the most studied or most well studied type of the CRISPR-Cas system, which is the type 2 CRISPR-Cas9. And so it has two essential components. So this CRISPR-Cas9, it has two important components. One is the guide RNA and another is the CRISPR-associated proteins or the Cas protein. So this guide RNA, so if you see, so this is the structure of the Cas9 protein in complex with the guide RNA, single guide RNA. Okay, so the Cas9, so this figure is taken from this book, Biochemistry, Leninger by Leninger. So the Cas9 protein, the first Cas protein extracted from this bacteria, Streptococcus pyogenes. That is why it is called SP-Cas9. So it is a large multi-domain DNA endonuclease. Okay, it is a DNA nucleus. Its main function is to cleave the DNA. So it will cut the DNA. It will cause double-stranded breaks in the DNA, and then so it is a large around around 1360 amino acids are present, and it has multiple domains in it. So there are two main domains which is present in the Cas9 protein. One is the recognition lobe and another is the nucleus lobe. Okay, so the recognition lobe is involved in recognizing the CRISPR RNA, or it is mainly involved in binding to this transcribed cRNA region, or the hairpin like structure that was present in the transcribed RNA. That is involved, that is recognized by this recognition lobe. And then there are two nucleus lobes. So one is this REC1, REC lobe. So these are the recognition lobes. One is the REC1 lobe and another is the REC2 lobe. So these, what will it do? Uh, so these are the recognition lobe, and another there are two nucleus lobe. NUC lobe, which is composed of RUVC, HNH, and protospacer adjacent motif interacting domains. So the RUVC and NA and HNH domains are used to cut each single-stranded DNA. Okay. So, so there are two nucleus domain. One is this RUVC and another is this HNH domains. So these will recognize the particular sequence of DNA, or sequence of DNA that has to be cleaved. So the dash domain cleaves the target DNA while the dash domain cleaves the non-target DNA strand. So there are two domains. One is this HNH domain and another is the RUVC domain. So one of these domains, So there are two nucleus domains. One is H and H domain, and another is the rho C domain. So the H and H domain will cleave the DNA strand, which has 5' to 3' polarity, and the rho C domain will cleave the DNA strand, which has 3' to 5' polarity. Okay, so answer is option C. So you can see here in this figure that was given in your lecture slides. So basically, this CRISPR-Cas9 system consists of two lobes in the Cas9 protein: the recognition lobe, which is called the REC lobe, and the nucleus or NUC lobe, nuc lobe, and the rec lobe. Okay, so the recognition lobe contains Rec1, Rec2, and domains responsible for nucleotide recognition. You see Rec1, Rec2, and another is these two domains, HNH and the RUBC. Okay. The arginine, then there is this arginine bridge helix, which acts as a linker between the RUBC and the Rec domain, playing a crucial role in initiating cleavage activity upon binding to the target DNA. So the nucleus lobe contains two endonuclease domain. One is the HNH domain and another is the RUBC domain. The HNH domain cleaves the target DNA strand, while the RUBC domain cleaves the non-target DNA strand. Okay, so the PAM interacting domain confers PAM specificity and initiates binding to the DNA. So the target DNA is the DNA which has to be cleaved. So that has a PAM sequence or protospacer adjacent motif sequence, which is present downstream to the target sequence. So that particular sequence is recognized by this PAM interacting domain. Upon DNA binding, the positively charged residues in the interface between Rec and nuc lobes, especially at the bridge, stabilize and neglect. So you can see this is the domain organization of this Cas9 protein. So there are multiple domains. There is roof C and this H and H, which are the nucleus domains, and then there are the recognition lobes, which are Rec1 and Rec2, and then again there is a PAM recognizing domain, which will recognize the PAM sequence which is present in the target DNA molecule. Okay, so yes, as I was discussing about the guide RNA. So the guide RNA is made up of two parts. One is this CRISPR RNA, and then there is this trans-activated CRISPR RNA. So this region, so CRISPR RNA is the RNA which is transcribed from the spacer sequence. So the RNA which is transcribed from the spacer sequence, that is called the CRISPR RNA. Okay, CRISPR RNA, and then there is a hairpin-like structure. So this hairpin-like structure is the tracer RNA, okay, or the trans-activating CRISPR RNA. So this CRISPR RNA, it is 18 to 20 base pair in length that specifies the target DNA by pairing with the target sequence. So this CRISPR RNA will be complementary to the target DNA that has to be cleaved. Okay, and along with it, there is the trans-activating CRISPR RNA. So it is a long stretch of loops that serve as binding scaffold for Cas9 nuclease. So this is acting as a trans-activating CRISPR RNA. This particular hairpin-like structure that is getting formed, that is recognized by the Cas9 protein. So for gene editing, the guide RNA is synthetically designed by combining this CRISPR RNA and the trans-activating CRISPR RNA to form a single guide RNA. So the single guide RNA will have two parts. One is the CRISPR RNA and another is the trans-activating CRISPR RNA. So these two will be you will have to design a plasmid having the sequence of both CRISPR RNA and the Cas9 uh, uh, Cris, uh, CRISPR RNA and repeat this repeat sequence, and along with it there has to be the gene for Cas9. So together this will express this Cas9 and CRISPR RNA. So what is the mechanism of action? So what will happen? The designed single guide RNA. The designed single guide RNA will it will direct the Cas9 and help in recognizing the target sequence in the gene of interest through its 5' CRISPR RNA complementary base pair component. Cas9 protein remains inactive in the absence of single guide RNA. So on its own, the Cas9 protein is inactive. Only when it will go and bind to this single guide RNA, only then it will know where is the target DNA it has to bind, because this spacer sequence will be complementary to the target sequence and it will provide the information where the Cas9 protein has to bind. So the Cas9 nucleus, once it is recognized, it is bound to this guide RNA, single guide RNA. It will bind to the target sequence and it will make double-stranded breaks at a site 3 base pair upstream of the. So this is the FAM sequence that is marked in this magenta color. So okay, so this PAM sequence is very essential, because if this PAM sequence is not present, then this CRISPR this complex ribonucleoprotein complex will not go and bind to this target DNA. Okay, so there has to be a PAM sequence. Okay. Two cursors and its size varies depending on the bacterial species. So the Cas9 protein recognizes PAM sequence. So this is 5' NGG 3'. So this is the particular sequence which is present in CRISPR-Cas9 system. So this Cas9 protein is activated for DNA cleavage. So what will happen? The H and H domain cleaves the complementary strand. Okay, the target strand, at the non-complementary strand will be cleaved by the roof C domain. Okay, through host repair pathways, mainly non-homologous end joining and homologous direct repairs. These cut regions can then be edited. Okay, so if you see this figure, this Cas9 and guide RNA, is forming a complex. It is going and bind to a particular region of the DNA, and then it is cleaving that portion of the DNA. And then this DNA is again repaired by the repair pathways which is present in the host. So what is the correct order of steps in the CRISPR process? So first there will be binding, right? First there will be binding of what? No, first no. So there is so first targeting will take place, right? Targeting means the spacer sequence or the this single guide RNA will uh, will uh, will determine where this CRISPR-Cas complex has to go and bind. Okay, so first targeting, then binding, then cleaving, and then repairing, isn't it? So it is option A. So second. Okay, now let's answer this question. Answer is option A for that. How does the guide RNA help the cell know where to cut the DNA? So how will the guide RNA help the cell to know where to cut the DNA? So the guide RNA has the spacer sequences, isn't it? So it will have the spacer sequences, and this spacer sequence is complementary to the DNA sequence, to the target DNA sequence. So answer is option B. Guide RNA is complementary to the DNA sequence, target DNA sequence, and that is why. So it is guiding the Cas9 protein, that at which portion of the DNA, where is the target DNA which it has to attack. Okay. The answer is option B. So this was discussed in your lectures as well. It depends solely on the Cas9 as the Cas protein along with two types of RNA, 3 and tracer RNA. This is precursor CRISPR RNA, and then these are cleaved into several short RNAs having the spacer sequence and the transactivating RNA. Okay. So now there are, as I was discussing, there are two types of CRISPR. One is the class 1 CRISPR and another is the class 2 CRISPR. So this class 1 is again divided into three types: type 1, type 3, type 4. And this class 2 is divided into three types: type 2, type 5, type 6. Okay. Class 1 and class 2. So what is the basis of dividing this CRISPR-Cas system into two classes? So they have been divided into two classes on the basis of whether multiple Cas proteins are involved or whether a single Cas protein is involved. So the class 2 system involves a single protein for interference. So there is a single Cas protein in class 2 system, while there are multiple Cas protein in class 1. Okay, so this taxonomic studies initially classified the CRISPR-Cas system into three main types based on particular marker proteins. So Cas3 protein is present in type 1. In class 1, there are three types: type 1, type 2, type 3. Then there are three different Cas proteins. Okay. To answer this question now, what is the major difference between class 1 and class 2 CRISPR? So just now I was discussing that the class 1 system utilize multiple Cas protein and the class 2 system involve a single protein for interference. So all these different classes and types have been discussed in your in the lectures very elaborately. So you can go through them. This answer is option C. Then there is another concept called dCas9. So dCas9 stands for the dead Cas or dead Cas9 protein. So this Cas9 protein has a mutation, which prevents the Cas9 protein from causing this double-stranded breaks. Okay, so this Cas9 protein will can go and bind to a particular sequence of the DNA, but it cannot leave the DNA. Why? Because it has the particular mutation. One is this H840A mutation and another is this G10A mutation. So it is a catalytically inactive protein. So the native Cas9 protein is a nucleus that has the power to cut either double strand or single strand DNA. But this DCas9 is specialized, artificially synthesized Cas protein that lacks one of the important properties, which is catalytic activity. So it is artificially synthesized and it lacks this catalytic activity. This nucleus activity is lacking in this protein, and the catalytic domain of this enzyme is modified in such a way that it keeps intact its DNA binding abilities but doesn't execute cleaving. So it cannot leave the DNA, but it can bind to that particular sequence of the DNA. So the decasing can be achieved by introducing a mutation into the catalytic domain, either D10A or H840A. So either mutation is designed for only preventing enzymatic activity but not DNA binding capability. So then why has this been? So if it does not have this nucleus activity, then why has this type of protein been designed or engineered? So it has been engineered because this it can be utilized or it has several applications. So it can be used in altering epigenetic activities, investing chromatin structure, base editing, epigenetic remodeling, and program chromatin interaction. So, for example, if there is a particular sequence of the DNA which is oncogenic or which is in a cancer cell, say for example, this particular gene has become very active and it is getting expressed. It is being overexpressed. So you can design the Cas9 system which can go and bind to this particular DNA sequence and inhibit the transcription of this particular DNA sequence. So such type of applications can be done with by using this dCas9 protein. Okay. So the dCas9 differs from the traditional Cas9. So D stands for dead Cas9. Okay, dead Cas. So how is it different from the traditional Cas9? So it has option is reduced binding specificity. This is wrong. So binding specificity is there because that is being conferred by the by this single guide RNA. Okay. So a lack of RNA binding capability, that is wrong. Enhanced DNA cleavage activity, that is wrong. Lack of DNA cleavage activity. So it has the DNA cleavage activity is lacking because of two mutations in the Cas9 protein. Okay. So it has two mutations. One is the D10A mutation and it's the H840A mutation that deactivates both the ROFC and the H and H nucleus domain, leading to nucleus dead Cas9 molecule. So one mutation is in this ROFC domain and another is in this H and H nucleus domain, which uh, makes this Cas9 protein incapable of causing DNA cleavage. Okay. Now we will quickly discuss the talent. Okay. I hope all these things were clear with respect to the CRISPR-Cas9 system. So it is basically there are a few points I have to remember. It is an adaptive immunity of the bacteria. Okay. It's an adaptive immunity of the bacteria, and there are for this CRISPR-Cas system. What is there? It is a particular basically CRISPR-Cas9. It is a specific sequence which is present in the bacterial genome. And how does this sequence look like? It has two main regions. One is it will have a Cas gene, so there will be a Cas gene, and along with this Cas gene, there will be a CRISPR array. Okay, there will be a CRISPR array. And what is this CRISPR array? So this CRISPR array again has two parts. One, in this CRISPR array, there will be a spacer sequence, and then there will be palindromic repeats. This will alternate, it will be alternately arranged. Okay. Spacer sequences. And what are these spacer sequences? These spacer sequences are unique. They're unique. While these repeats are, they're identical to each other. These repeats, they're similar. They're identical to each other, but these spacer sequences are unique sequences, and these spacer sequences are sequences that have come from the genomic sequence which is present in the bacteriophage. It is actually a memory system of the bacteria, and how we are engineering it. We are so this this Cas gene and this array having this spacer can be designed, and it can be create a construct, plasmid construct having this Cas gene and this CRISPR array, and then you can transfect it in a mammalian cell, and this spacer sequence will just give me a minute. Now. Okay. So this spacer sequence will is complementary to the target DNA sequence, which has to be cleaved. Okay. So now let's talk about talent. So talent is another genome editing system. So this talent, so this stands for transcription activator like effectors, effector nucleases. So this talent again, it comprises a non-specific forky fork1 nucleus domain fused to a customizable DNA binding domain. So you see, this is the structure of a talent. How a talent looks like. So it has four main domains. One is the N-terminal domain. Then there is this tail repeat domain. Then C-terminal domain and this fork1 nucleus domain. So this fork1 nucleus domain will code for this fork1 protein, as we had discussed in zinc finger nucleases also. So the zinc finger nucleus also had this fork1 nucleus domain, and this will form a dimer and cleave the particular sequence of DNA, which the sequence which will be decided by this tail repeat domains. Okay, so this DNA binding domain is composed of highly conserved repeats derived from transcription activator like effectors, which are proteins secreted by Xanthomonas bacteria to alter transcription of genes in the host plant cells. Okay, so this DNA binding domain, so this is where this protein or this protein will go and bind to the DNA that is conferred by this transcription activator like effector. So you see here these, you see HG, NN. So we will understand what are these. So these are basically these amino acid residues uh, will goes for a particular is Okay, so these are the tail repeat domains. So what are these? So the fundamental building block that is used to engineer the DNA binding regions of talents is a highly conserved repeat domain derived from naturally occurring tails encoded by Xanthomonas species protobacteria. So these tails are injected into host plant cells via type 3 secretion system and bind to genomic DNA to order transcription in host cell, thereby facilitating pathogenic bacterial colonization. So the DNA binding is mediated by arrays of highly conserved 33 to 35 amino acid received repeats that are flanked by additional tail-derived domains at the amino terminal and carboxy terminal. So I'll explain it in the next slide with the help of a figure. So you see here, so the individual tail repeats in an array specifically bind to a single base of DNA. So this repeats, HD repeat. So you see here, these are the DNA binding repeats. So this DNA binding repeats is composed of 35 different amino acid residues. And out of this 35 amino acid residues, the 12th and the 13th amino acid residue is different. All other residues are the same. So this 12th and 13th residue will decide that what particular nucleotide base it has to go and bind. Okay, so that is what is explained here. So the individual tail repeat in an array specifically bind to a single base of the DNA. So which repeat decides which base it has to bind. So that is done by this 12th and 13th residue. Okay, so a thymine is conserved at the position just 5 prime to the base. So always there will be a thymine. Okay, this thymine is always conserved, but the other sequence, so where it has to bind, that is decided by what is this particular repeat which is present in this tail binding domain. Okay, so the tail repeat arrays published to date use 4 domains that contain the hypervariable residues. So these two residues, this 12 and 13 residue, these are called the hypervariable residue. So they are NN, NI, HD, NG. Okay. So this, if there is NN hypervariable, so if the 12th and 13th residue is NN, then it will this will this will indicate that this protein has to go and bind to the guanine base. If it is Ng, it will give it specificity that it has to go and bind to the thymine base. So in this way, so NN stands for the recognition, for recognizing the guanine nucleotide. Ni will help in recognition of the adenine residue, Hg for cytosine residue, and Ng for thymine residue. Okay, so these tails are transcription activator like effector protein. So this is the domain organization of this protein. It has a nuclear localization signal. Okay, so it is a nuclear localization signal. Then there is this N-terminal localization signal, TS. So you can see these are marked over here. NLS is nuclear localization signal. Then there is a N-terminal translocation signal. This is the translocation signal, transcriptional activator domain, ET, and central repeat domain. So this is the central repeat domain. The primary distinguishing features of transcription activator like effectors is a central repeat domain. So this is the central repeat domain, which consists of 1.5 to 33.5 repeats, each typically 34 residue long. So these are the repeats. Okay, so this is this is made up of 34 residues. This is again made up of 34 residues. This is again made up of 34 residues. So in this 34 residues, only 2 residues are different, which is called the hypervariable domains. So similarly here also the 2 residues are domain, the 12th and the 13th. So this 12th and the what is the 12th and the 13th residue will determine which is the particular nucleotide that will be recognized by this talent protein. Okay. You see here, this has been exercised and what I was explaining has been shown in this figure also. So these are 34 different residues and the 12th and the 13th residue are hypervariable. So these are hypervariable. So these are also called the variable die residue or RVD, variable high residue. And the identity of these 2 residues in consecutive repeats corresponds to the sequential DNA bases in the TALS effector's target site. So this corresponds to so if this is as was explained here, and show this figure. Okay, so if it is NN, then if it is NN, then it will indicate that it will go and bind to. So for example, I've shown this in this figure. In NN, it means it will go and bind to guanine. So if this is, so the what were the four domains? NN, NI, HD, NG. NI, then it will NI, NG. So these will say that which is the particular nucleotide base where it has to go and bind. So you can engineer this. So what will be these hypervariable domains that can be designed, right? Depending on your target sequence, you can keep what will be the corresponding hypervariable domain that you have to that has to be present in this in this talent routine. So now answer this question. So this is the last concept that was discussed. So this last question is the talents are created by combining a TAL effector transcription activator like effector DNA binding domain with a dash which acts as a nucleus capable of cutting DNA strand. The answer is so just like zinc finger nucleus also here also it is the same. It is a DNA domain which is used with a helicase domain. Not helicase, it is DNA cleavage domain. DNA cleavage domain. So the TAL affected DNA binding domain will will determine or it will provide this TALEN molecule where it has to go and bind, and this nucleus will cleave that part of the DNA. Okay, so the TAL DNA binding domain, TAL effectors. TAL effectors, the DNA binding domain comprises a repeated 33 to 34 amino acid sequence with variable 12th and 13th amino acid known as repeat variable diresidue. Repeat variable diresidue, RVD. So the RVD variation correlates strongly with specific nucleotide recognition. Okay, allowing the engineering of customized DNA binding domains by selecting repeat segments with the desired RVDs. Minor alterations in the RVDs and the incorporation of non-conventional RVD sequences can enhance target specificity. Okay, and then there is the non-specific DNA cleavage domain, which is the foci endonuclease. So the foci endonuclease, this forms a dimer and cleaves that particular sequence of the DNA, which is specified by this TAL DNA binding domain. Okay. To construct hybrid nucleus, the foci domain operates as a dimer. Requiring two constructs with distinct DNA binding domains for target genome sites with appropriate orientation and spacing. The distance between the TAL DNA binding domains and the foci cleavage domain, as well as the spacing between individual TALEN binding sites, crucial factors. I hope all these concepts are clear. You see here, so this is a dimer. So here in this figure also you can see, this is the foci endonuclease. The foci endonuclease, this forms a dimer and will cleave that particular region of the DNA. Will cleave a particular region of the DNA, and these are the DNA binding domain. So this hypervariable domain is allowing the recognition of a particular nucleotide. This is recognizing a particular nucleotide. This is the way you can in which these talents are designed. Okay, so that was all for this session. So tomorrow will be the last problem solving session for this course, and um, I hope this session was useful. The remaining part we will take up in the next class in the week 12, mainly the week 12 like concepts will be discussed tomorrow.